This isn't, you didn't start from ground
Absolutely.
zero in April. You took something already in existence that's been looked at for years, and starting in April, you began the process to look at it in context of COVID-19.
Absolutely. We've done 25 years of research into this compound, and it's a first-class scientific team. We have lots and lots of very serious work on the effects of this compound, both in cancer and now in viruses, and with some very, very promising data and more than 20 published papers on the topic. So the idea that we somehow haven't followed any process or there's no support for this is ridiculous. Secondly, there's this idea that it isn't safe. Well, we've already talked about that. We've already taken it through phase 1 and phase 2 clinical trials in humans with the FDA, and at dosages up to 16 times the proposed dose, I might add. So that's, the idea that it's not safe is absurd. You'd have to take 600 doses of our proposed product to suffer any negative effects from it. Nobody's going to do that. The third thing as well is that there's no proof that this could in any way be efficacious or work. Well, that's just not true either, because we've already done a study with UTMB in Galveston where we showed in monkey liver cells in a laboratory an average 20,000-fold reduction in viral infectivity in the first 24 hours. That's news. Yes, it's in the lab, but that's still news. There was also some initial proof of prophylaxis as well. That is in a paper that has been published. So that's out there too, and you can read that. It was going through the peer review process, but with the that kicked off now, the journal that was going to publish it is now having second thoughts about doing a peer review on it due to the amount of pressure they've seen over this, which is remarkable.
They're trying to keep people from seeing action. Now tell us how, the basic idea of how it works. You mentioned it to me about the envelope. Tell us about that.
So in the course of our research, one of the things we've discovered is that in the presence of our product, the glycoprotein wrapper around the RNA strand in envelope viruses is malformed and or missing certain glycoproteins. That seems to have an effect on the efficacy of the virus. So there are a great many factors that could explain how that occurs, but nevertheless, we've actually seen that in a number of different circumstances and published material on the topic. So that we call that our envelope theory. Now, a number of viruses are envelope viruses, and coronavirus, thankfully, is one of those viruses. So that's what led us to UTMB in the first place. They've tested over 53,000 compounds, Kevin, and ours is one of those. They also did a lot of the work on remdesivir with Gilead.
Right, and so when we're talking an envelope virus, if I understand it correctly, and again, I'm not a doctor, but there's this protective fat wrapping around the virus that kind of protects the virus, and when we wash our hands, we break down that fat, that envelope, and so it kills the virus off our hands. That's why we use soap to wash our hands. Alcohol does something like that too, but we're talking here about something that you all have been testing in relation to it can be consumed by people, and it, like other drugs, could potentially break down that envelope, and that might be why it works, right?
It could well explain why it works, Kevin. What we can say definitively is an average 20,000-fold reduction in viral infectivity when you measure the virus progeny. Because a virus enters the cell and essentially hijacks the cell and uses it to make more itself, kind of like a photocopier out of control. The problem is the cell itself is then not doing the job it should be doing, which is why you feel the detrimental effects of the virus. The virus progeny particles then go on to infect other cells. So if our compound in a laboratory setting can drive a 20,000-fold reduction in viral infectivity, that's big news.
Huge.
The next step, obviously, is to test that in animals and humans. Well, the animal testing is underway, and what we're looking for is the opportunity to do human testing under an IND in a controlled set of circumstances. That is all we're asking for. And no more, no less.
And that's what we've got to get the word out on. They're trying to kill a potential, strangle the baby in the crib is the kind of idea. Kill a potential treatment before it gets looked at, because it's not a highly expensive treatment. It's something that's all natural. It's something, they didn't circumvent the process. They did not violate FDA protocols. They followed those. They did not try and ask the president to do something untoward, and he didn't offer to do anything untoward. Secondly, it has not been proven to be dangerous. They're over the top in trying to tell you, well, it's poison. It's made from poison. A lot of the drugs that you take are made from poison. And the third thing is it has shown tremendous promise and deserves to be looked at. So I think we're going to start a petition that the media back off and start allowing something as promising as this to at least go through the proper process. Andrew, I think you're doing a great service for America in bringing this out, and I'm sorry for how the media has used a narrative to try and hit the president and trying to keep the American public from learning something promising, and they've used it to attack you, your family, and your colleagues.
Hey, Kevin, I appreciate the opportunity to talk about it. We're all here for the same reason. We just want to get this country better, get the country back on its feet so we can all go back to our normal lives before this virus hit us. The reality is we have something that's all natural. It's available in a plentiful supply right now in enormous quantities, and we would like to bring it to bear. Obviously, we need to do that in a safe setting. We've already gone through phase 1 and 2 clinical trials in humans, so we know it's safe. There's no question about that, and therefore there's absolutely no downside to allowing us to test it. Where's the harm in that? Call our bluff. Let's see.
Well, you know, we want our viewers to be able to listen to those actual interviews that you did so they can see the truth and see how things were twisted and manipulated. But we also want them to have access to all of the data that you have. Where can people find, we're going to put it on our website, we're going to put it in our battle plan, but where can people find more about this?
You can go to the company website, phoenixbiotechnology, all one word, .com.
phoenixbiotechnology.com. All right, thank you.